All right, good afternoon, everyone, and welcome. It's my pleasure for today's NHGRI seminar series to introduce Rasmus Nielsen from Berkeley. Rasmus has spent most of his career studying using genomic tools and developing those tools for studying evolution, particularly in terms of human adaptation, but also I just found out, interestingly, other areas of interest, including poison frogs, which I'd like to hear the poison frog talk someday as well. So Erasmus went to graduate school at UC Berkeley, working with Montgomery Slatkin, did his postdoc at Harvard, brief stint at Cornell, and then moved to the University of Copenhagen, and now at UC Berkeley, where he has a joint appointment in those two places. I will give a congratulations. His impact on this field has been significant for a long period of time, and that's resulted in him being just recently elected to the National Academy of Sciences. And so we're looking forward to a very exciting talk from Rasmus today. Please join me in welcoming him to this podium.
Thanks so much. Thanks for the introduction. So I'm gonna talk a bit about evolution today. You might ask, okay, why should we be interested in studying evolution? Well, a lot of the time when we study evolution, it's for the sake of evolution itself. We might know something about something functional, some functional relationships, the action of some transcription factor, how it works, and we want to know how it has evolved. But sometimes we can also go the other way around. We can study evolution, and from that we can learn something also about function. And I'm going to give some examples of that today, where if you're more on the functional side, keeping an eye on or being aware of evolutionary biology might actually sometimes help you a little bit as well. So as evolutionary biologists, we like to divide selection that works on the genome in two different categories. One is negative selection. That's the idea of a new mutation that enters the population, and then with high probability it will be lost from the population because individuals that carry that mutation, they have lower fitness, they have a disadvantage because they carry that mutation. It's a deleterious mutation. So if you do bioinformatics, looking for negative selection, that's one of the main tools that we use to understand function. We look at what is conserved. We compare, for example, different mammalian genomes with regard which regions are conserved. And we know because they're conserved, that must be because they're functional. So when we're doing that, we're really being evolutionary biologists. We're making an assessment that negative selection is acting in that genome to weed out deleterious mutations, and therefore it's functional. So in that sense, everybody who has ever done anything bioinformatically, where they take advantage of any kind of annotation that might rely on what is conserved, they're evolutionary biologists. And I'm not going to really focus on that kind of selection much more. Mostly what I'm going to talk about is something that we then do less, perhaps. That is positive selection. That's the opposite situation. We imagine a mutation that enters the population, gives individuals who carried a fitness advantage. And of course, people or individuals that carry that mutation have a fitness advantage with high probability. Eventually, it will go to fixation in the population. So the reason why we're interested in those kind of mutations is because they're the substrate of evolutionary change. If we want to understand adaptation and how organisms evolve and diversify, this is a type of selection that you're going to look at, the type of selection that creates new function. But there's also, in the context of human genetics, another reason that we're interested in it. In particular, if we're interested in mutation that might have a strong effect and be common in the population, that's the kind of selection we're going to look at. So most of you are probably familiar with this type of plot that resulted from the genome sequencing, the GWAS era, and so on. We were able to find mutations that were common, that were high frequency in the population, but with a very small effect size. We found many of those for complex diseases. We also found mutations that are very rare and have a strong effect. So those will be the kind of mutations that affect, say, rare Mendelian diseases, for example. And then something in between. And of course, we think there are lots of mutations out there of rare variants with small effects that are not very interesting. But these type of mutations, common mutations that have a strong effect, it turned out there weren't very many of them. If we're going to understand common diseases like cardiovascular disease, type 2 diabetes, cancer, and so on, generally these diseases could not be explained by these type of mutations. There weren't very many of them. And there are good evolutionary reasons why you wouldn't expect to see them. If there are mutations that have a very strong effect on your health, you wouldn't expect them to have a really high frequency in the population. You would expect them to have been weeded out by natural selection. So we could maybe have told ourselves before we started sequencing genomes that we shouldn't find many of these. But there's one exception from that, and that's new mutations that enter the population and carries a fitness benefit to them. For example, because individuals have changed their environment. And so those are these mutations that are under positive selection. And what I'm going to argue here today is that we actually have numerous examples of this in humans, of mutations of strong effect, of high frequency, that exist in various populations because they've been under positive selection. And we cannot replace DIR studies, obviously, by looking for these mutations, but it gives us an extra opportunity to find, in some cases, mutations that are common in the population of strong effect. So before I get into detail of some examples of that, I'm going to spend about 10 minutes to talk about what we really spend our time on, that is the computational and statistical side of this methods, development for finding, for example, this type of selection. And if you're really not into computation and methods, feel free to take a nap for the next 10 minutes and then wake up again when I talk about some examples. But I'm going to spend just a little time to talk about some of the computational methods that we are developing and using. So what we're looking for when we're looking for this type of selection, positive selection, we're looking for the effects of a selective sweep. So imagine we have a population here with some different DNA sequences. These strings here represent DNA sequences. There's some mutations on them in the population, and then you have a new mutation that arises here. And if that mutation is advantageous, it may increase in frequency in the population, and you might end up with a situation like this, where then all of the individuals in the population, they carry that mutation. And the important thing here is that it's not just that mutation that increases in frequency, it's all the other linked mutations. So there will be a specific pattern in the genome that we can look for of this type of event. That's called a selective sweep. And we have spent the past, part of what we've been doing for the past 20 years has been developing more and more refined methods for trying to find these selective sweeps. So what we're doing right now is that we're using ancestral recombination graphs for this. And I'll just going to spend a few minutes introducing this idea about ancestral recombination graphs for you if you're not familiar with that idea. So an ancestral recombination graph is a graph that in it has all of the evolutionary trees, the trees that relate individuals to each other in all positions of the genome. You may be familiar or have heard about a coalescence tree. That is in a particular position in the genome, or for maybe mitochondrial DNA, you can make a tree of the individuals in your sample, the coalescence tree. Because of the process of recombination, that coalescence tree will change along the length of the genome. So in the human genome, you will have millions of different coalescence trees. The collections of all of those trees and the way they're connected together by recombination events, that's the ancestral recombination graph. And we have statistical theory that tells us that all of these processes we're interested in, the effects of mutations, the demographic history of the population, and natural selection, that's all embedded in the ancestral recombination graph. If we have the ancestral recombination graph, we have all of the information that's worth having about the DNA sequences that we have. So the key to make inferences these days from full genomic data is to get at this ancestral recombination graphs. And fortunately, we're getting better and better and better methods for estimating these ancestral recombination graphs, and therefore for getting all of the important information, for example, about selection, out of the data. So one of the methods we are working on these days, and we have developed and have used for some of these studies, is based on these ancestral recombination graphs. And what the method does is that it uses methods for sampling ancestral recombination graphs according to their probabilities. And what I mean by that is that instead of just getting one ancestral recombination graph, you get a cloud of them that represents all the uncertainty about ancestral recombination graphs. At the same time, we also get a cloud of trajectories, allele frequency trajectories of each mutation back in time. These are two latent variables. So these are variables we can't observe, the ancestral recombination graph and this path of the allele frequency. And what we do is we integrate That is, we sum probabilistically over all the possible paths of all mutations informed by all ancestor recombination graphs. So that's quite computationally intensive, but it is possible to do, and something we've been doing for selection and also for other purposes. So there's two hidden layers here. We have a hidden layer that is the ancestor recombination graph, and another hidden layer that is the allele frequency trajectory for each mutation. So what we can do with this framework is we can not only get all these ancestor recombination graph, but we can get probability distribution of what the allele frequency was back in time for all the mutations in the genome. So the result of this is something like this. We have here time in the past, and then this is a specific mutation where we have the allele frequency back in the past. And these are from some simulations, the reds. That's the true allele frequency back in the past, back to 5,000 generations ago. And then the colors, that's our inference of this, the path, what it was, and the associated statistical uncertainty around that. So this is a case where there was no selection acting on the mutation. It didn't change allele frequency much because there was no selection acting on it. This is a case where there was positive selection acting. So back in past, if you look back in the past, the allele frequency is decreasing, meaning that looking forward in time, the allele frequency was increasing because of this positive selection. And again, the red is the true value in our simulations, and the yellow represent our estimates. So we can estimate these allele frequency back in the past, and with that we can also estimate how much selection was acting on the mutation. And it's quantified through a parameter that's called the selection coefficient. That tells you something about the fitness of an individual that carries the mutation versus the fitness of an individual who does not carry the mutation. If S is large, there's a strong effect of selection. We can get a likelihood surface that can quantify information about it, and we can estimate the selection coefficient in the cases where there is selection and get relatively precise estimates, and when there was no selection. All right, and from that we can then, and maybe I won't go through the technicalities of this, we can simulate and show that this is a very powerful method of finding where in the genome has there been natural selection acting. We get all the information in the DNA sequences about the allele frequency in the past of the mutation, and that gives us all the information about this selection coefficient. So this is an example from the lactase locus. You might be familiar with the story of lactase. So several different places in the world, individuals in cultures where they have dairy farming, they have evolved lactase persistence. That is the expression of lactase as adults, so you can digest lactose, the major carbohydrate in milk. And that's happened several different places in the world. This is for some Europeans, where we have here some sequence from some DNA sequences. We estimated the frequency in the past through this black line here of the allele that confers lactase persistence. And in red, we have results from another study that used ancient DNA to get the same allele frequency in the past. They differ a little bit, and we think that's because of the slight differences in the populations that we're looking at, but basically it's the same conclusion. And sort of a verification that we can, with modern sequences, basically estimate the same thing as you get if you have actual ancient sequences in the past. We can estimate these trajectories back in the past. So this is a result for a bunch of different loci that are associated with pigmentation in humans and in Europeans, and been all argued to be associated with selection. And we can see in some cases we get strong evidence of selection. For example, this locus here, KITLG, KIT ligand. There's been an increase in frequency of the allele in the past, and we get an increase, high selection coefficient, about 1.6%. It's a very strong selection coefficient in humans, and the evidence is in terms of this log likelihood ratio of about 30 here. And then we get some other cases where the people argue there was selection, where we can show, okay, actually there's zero evidence in the data that there's been selection in the past for this particular allele. So we can go in the genome with a set of DNA sequences, and we can quantify how much selection there is and which loci there's been selection. Now, part of the trouble that we've had with this kind of thinking that we've been doing for many years is that we look at selection on a single SNP. How much selection is on the SNP. And as you know, as geneticists, connecting a SNP to a phenotype is a very complex thing. In particular, almost every SNP that has a strong effect on one phenotype will affect other phenotypes too. Similarly, each phenotype is affected by many different SNPs, typically. Almost any trait we're interested in is highly polygenic. So we can take advantage of that and then look at selection that acts on the phenotype instead of selection that acting on individual SNPs by combining the information from multiple SNPs. So we've done that. We can do that using quantitative genetic theory, and again, we can show that we have methods that have a lot of power to detect selection not acting on a SNP, but on a trait. Now, just to show an example on that, this is from humans from UK Biobank data. We look again in Europeans here, look at which traits has been under a lot of selection. And what we find is that a trait such as tanning, for example, has been under a lot of selection. So what is tanning? That's something you ask people their ability to tan. That is to develop skin pigmentation, and not surprisingly, in Europeans, we find on that trait that has been measured this way, we find a lot of selection. Many other studies have shown that skin pigmentation has been under strong selection in Europeans over the last 10 to 20,000 years. Zooming in a little bit here, we can see some of the other traits that has been associated with strong selection in Europeans. Now, the challenge here with this type of inference is, as I mentioned before, also that for each SNP, it affects multiple different phenotypes. We have a high degree of pleiotropy for almost any trait that we'll be looking for. So if I say that, oh, okay, a bunch of SNPs, this SNP and that SNP and some other SNPs have been changing in allele frequency, and we think it's because phenotype 1 has been under natural selection. It's often very hard to show that it's not phenotype 3 that's been under selection, because they may be affected by the same SNPs. If you have a set of SNPs that strongly affect the phenotype, that many of them likely also to affect other phenotypes. So how can we get around this problem? Well, we can quantify the genetic correlations. Because we know which SNPs are associated with these phenotypes, at least between the phenotypes that we have, we can quantify the genetic correlations between these phenotypes. And we can incorporate that into the statistical framework. And by doing that, by incorporating these genetic correlations, we can ask the question of, was selection actually on one trait, or was it the other trait it was actually acting on? I'm just going to show one example of that. Again, going back to skin pigmentation. There's been a lot of debate about hair in Europeans, because Europeans not only have had a change in skin pigmentation, they also have hair pigmentation, different hair color. And for skin pigmentation, we think we know what's going on there. It has to do with vitamin D deficiency. If you're in an area where there's less UV radiation, you will suffer from vitamin D deficiency because you need UV radiation to synthesize vitamin D. Going into a climate where there's very little UV exposure, you will suffer from vitamin D deficiency, and that's supposed to be what drives the change in skin pigmentation in Europeans. But then what about hair color? There's been lots of hypotheses about various forms of sexual selection, all kind of ideas about why hair color might have been under selection. Many studies have argued that hair color have been under selection. But it turns out actually that, oops, that's one too much, that if we look at hair color and then condition on skin pigmentation, there actually is almost no evidence for selection on hair pigmentation. So this is the p-value we get for pigmentation associated with the hair color, the evidence of selection, if we don't take skin pigmentation into account. But if you take skin pigmentation into account, this is the p-value that we get. We can explain almost all of the signal relating to selection on changes in hair color in Europeans by the changes in skin pigmentation. Clearly, many of the loci that controls pigmentation in the skin also controls pigmentation in the hair, and the changing hair color might just simply be a pleiotropic effect of the selection for changes in skin pigmentation in Europeans. All right, so that was a little bit about the sort of type of methods that we're using right now. In the past, we used all kind of other different methods for trying to detect selection, and I'm now going to just give you a few examples of how we use this. So look in humans, we also look in many other organisms, but looking now here at the NHGRI, looking in humans to try to detect selection in humans, particularly this type of positive selection. And the first project I'll talk about is Tibetan altitude adaptation. So the Tibetan plateau is in an altitude of 4 kilometers, and that means that the partial pressure of oxygen is only 60% of what it is at surface level. So that means with each breath of air you take, you only get 60% as much oxygen as you would get at surface, at sea level. Of course, then you will suffer from oxygen deprivation, from hypoxia, and there's a physiological response to that that includes an increased production of red blood cells. So if you're brought up at sea level and you go to the Tibetan Plateau, part of the response to that will be that you start producing more and more red blood cells. So that's a good thing because it increases the transport of oxygen in your blood. But it also has an other effect, of course. Your blood changes its viscosity. It becomes thicker. And that has a bunch of negative fitness consequences. For example, it increases the chance of stroke. There are also very strong pregnancy complications associated with the increased blood viscosity, and something that both in the medical and anthropological literature has been studied for many decades, this effect, the negative fitness effects of what you could say is an overproduction of red blood cells in high altitude. Now, what's special about, to fast forward here, what's special about Tibetans is that they don't have that response. In high altitude, Tibetans, they don't overproduce red blood cells. They have a slight increase in red blood cell production, but not anything similar to other populations. And it's supposed to be an adaptive response, that they are adapted to the life in high altitude by not overproducing the amount of red blood cells. And one of the things that we did was we then searched using some of these methods for trying to detect selection. We searched for loci associated with that. And we were able to find this locus, EPAS1, as the strongest outlier with a very strong signal of natural selection in Tibetans. So what is EPAS1? Well, it is a transcription factor which regulates the response to hypoxia. So it's a very good candidate gene. We also found one other candidate, EGLN1, which is also in the same hypoxia response pathway. And we were able to, using some very simple association studies, to show, in fact, that much of the difference between Tibetans and then Han Chinese that we compared to in red blood cell concentration in high altitude could be explained by our lead SNP or something in linkage disequilibrium with this lead SNP. So that it seems that much of the response, the unique response of Tibetans to low oxygen levels, to hypoxia, is in fact caused by this gene EPAS1. So it's an example, as I talked about before, of a mutation of very strong effects of high frequency. And the reason why it has high frequency here is that it's been under positive selection in the Tibetan population, and it has lots of physiological effects. So you now have a mutation that is of high frequency and of strong effect because it's been under strong selection. Now, the next thing that we did was we're trying to say, okay, now we have a mutation like this. Let's try to model how selection works and try to understand that better. But the problem we were running into was that none of the models we have of how selection works could actually explain the pattern we saw in the data. So it's illustrated by this figure, the problem we were running into. So the way to read this figure here is that each line here is a haplotype, and each column is a SNP. And when it's black, that means it's the derived state. When it's white, that's the ancestral state. And then we have some individuals here that are Tibetans, and then Han Chinese individuals that we compare to down here. And you could just, from looking at this, you could see that there's a very big difference in many, many SNPs. We have basically two haplotypes that are very different between the Han Chinese and the Tibetans. And the problem that we have is there are too many differences here. Just because selection has been acting on a SNP, you wouldn't expect this many differences between the Han Chinese and Tibetans. We couldn't really explain that. So we have worked together with Svante Pääbo previously, who won the Nobel Prize last year, for showing that there has in fact been interbreeding between humans and Neanderthals, and for people who trace their ancestry outside of Africa, about 1% to 2% of their genetic material comes from Neanderthals. So we've previously been working on that. We're well aware of that. So we tried we asked the question, well, this divergent Tibetan haplotype, could it be Neanderthal? But it is not. It was in fact not Neanderthal. There was no match to that. Now, turns out that Svante Pääbo, as he was working on this Neanderthal genome, he got a little finger bone from the cave in south central Siberia, the Denisova cave. A little finger bone that the anthropologist there said, maybe this is also Neanderthal, and you're welcome to grind it up and extract DNA from this in your quest to sequence more Neanderthal genomes. So Svante Pääbo did that, but what he found was that it was something completely different. Oh, no match with Neanderthals. It was something completely different, and here we have the little finger bone here. It was something completely different than Neanderthal. It was something that we now call Denisovan, which is a very, very distant cousin from Neanderthals, almost as closely related to humans as the Neanderthals, but still on the Neanderthal lineage. We now call them the Denisovans. And when we compare to that, to this Denisovan sequence, we found an almost complete match. So the Denisovan sequence is a green button up here, and every place there's a match to the Tibetan sequence, you have a little star here. So it seems that the DNA sequence of EPAS1 that confers the different regulation of red blood cell production in the Tibetans, that was actually something that had been integrated into humans from these Denisovans. It's an example of what we call adaptive integration. Integration, that's the transfer of DNA through interbreeding between one species or another. Adaptive meaning it gives a fitness advantage. And we have now numerous examples in humans of this type of adaptive integration from either Denisovans or from Neanderthals. All right, so that was the story about adaptation in Tibetans. I'm going to talk a little bit now about another study we did working with Inuit from Greenland and health authorities in Greenland on trying to look at adaptations in the Inuit population to their lifestyle, which involves two factors. First of all, of course, it's cold in the Arctic. I should say the Inuit are the indigenous people that live in Greenland. They also live in Alaska and other places around the North Pole, areas and regions around the North Pole. And anthropologists have argued that they adapted to cold, perhaps, and then they also have a very unique diet that's really rich in fat and protein, especially polyunsaturated fatty acids, long-chain polyunsaturated fatty acids. And there's been a lot of interest in this. And it goes back to the 60s and 70s when first there was a lot of awareness that eating a lot of animal fat is bad for your cardiovascular health. And that made then some researchers ask the question, well, on a traditional Inuit diet, which is almost only animal protein and fat, if animal fat is bad for you, and they eat a lot of it on their traditional diet, the Inuit, how come they don't suffer from cardiovascular disease? The Inuit, on their traditional diet, suffer very little from cardiovascular disease. And to the idea that it's because of the kind of fatty acids that they're eating. They're eating these long-chained polyunsaturated fatty acids that perhaps are protective for your cardiovascular health. And if you go to a supermarket now and look at various supplements, you'll find that it's filled with these omega-3 supplements. And that's basically originally comes from this idea that omega-3s, these polyunsaturated fatty acids, long-chain polyunsaturated fatty acids that you find in, say, seals and whales and in fish, that they are in fact protective against cardiovascular disease, and later on it's been claimed also other diseases, cancer and other things. And it becomes one of the biggest dietary supplements in the United States. You can get milk that has now supplements of omega-3s in it as well, and so on. It all comes originally from this story about the Inuit and all the animal fat that they are eating. That is this animal fat from marine sources, long-chain omega-3 polyunsaturated fatty acids. Okay, so that was part of the interest. They have this unique, traditionally, this very unique diet, and we're interested in maybe there's also some unique physiology here. So we worked together with the Greenland health authorities and sequenced genomes for 2,500 Inuits. And then we did a scan for selection in the genome to try to figure out where it has natural selection been targeting the genome. And at the top peak, where we find the strongest evidence for natural selection in the Inuit, is a cluster of genes, FATS1, FATS2, and FATS3. So FATS3 we don't know that much about, but FATS1 and FATS2 we know a lot about what they're doing. They are they encode enzymes that catalyzes the rate-limiting step in the endogenous synthesis of polyunsaturated fatty acids. So this is the omega-6 polyunsaturated fatty acid synthesis chain, and this is the omega-3 polyunsaturated fatty acid synthesis chain. So you go from short chain to long chain polyunsaturated fatty acids. The short chain you would tend to get through a plant diet. The long chain one over here, you would get that from a meat or fish diet. And the idea here is that if you get fewer, if you have a more vegetarian diet, then you would want to have the synthesis of the long chain to work faster for you. If you have an animal diet rich in animal polyunsaturated fatty acids, a fish-based or meat-based diet, then the synthesis pathway should be slower. And that's exactly what we were able to see when we look at the Inuits. They had changes in these FATS1 and FATS2 genes that slowed down this endogenous synthesis of the polyunsaturated fatty acids. So they would accumulate more of the fatty acids that they otherwise would have gotten for a plant diet, and they produce fewer of the ones they would get from a fish-based or meat-based diet. So the Inuit have adaptive changes that in some sense compensates for the high intake of the long-chain polyunsaturated fatty acids. They accumulate more of the short-chain ones, and they endogenously synthesize less of the long-chain ones because of these mutations. Now, these mutations have a massive effect on almost anything you can measure. So this is LDL cholesterol. These are 3 different genotypes, GG, GT, and TT. These are the effect sizes on LDL. These are massive effect sizes on LDL cholesterol. Here we have to left mean weight. So there's a difference of when we do the statistics properly, there's a difference between of about 4 kilograms in mean weight from just one mutation. Okay, so that's a massive effect, way more like if you look at Europeans and all the big databases, cohorts there, there's a much bigger effect than you find for any common mutation there. This is a very common mutation, high frequency mutation in the Inuit. We have this very, very strong effect mutation. And also, oddly, has a strong effect on height. Okay, so this was a puzzle to us. Why does it affect height as much as this? Well, almost 2 centimeters in average. Well, it turns out that growth hormone, the regulation of the secretion of growth hormone is regulated by the composition of plasma-free fatty acids. That's something that's been known in the literature for a very long time. And it's something that is taken advantage of, for example, by chicken farmers. So chicken farmers know that if you add fish oils to the feed, the chicken become larger. Even though they get the same amount of fatty acids, the same caloric intake, they become larger if you add fish oils to their feed. And that presumably is because that triggers, even in chicken, a release of growth hormone. So we think what's going on here is that the Inuits, they have this diet that's rich in animal fat in their original diet. And that has posed this selection on these fat genes that causes many different physiological effects, reduce LDL cholesterol levels. It also has a massive effect on reduced fasting insulin levels. I didn't show that. We also think, and this is what we hypothesize, that there's this change in hormonal control that leads to reduced height. So that's our hypothesis about that. But one thing we can say for sure is that the lessons from the Inuit regarding polyunsaturated fatty acids cannot be extrapolated to other populations. That just because it's healthy for the Inuit to eat a diet that's very rich in polyunsaturated fatty acids doesn't mean necessarily it's good for other populations. And in fact, there's been a lot of studies coming out recently that's arguing that there's very little, if any, positive beneficial effect, at least on cardiovascular health, on these omega-3 supplements. It's highly debated, but it seems that in most meta-analysis that there's very little effect of that. Highly debated topic though, but the original motivation for it in terms of the Inuit health certainly doesn't hold up if you want to try to move those lessons to other population groups. All right, so that was the Inuit study, but just sort of a little, one other little observation here. We also have this other peak here that we see, where we think the selection associated with a gene called TBX15. And while we could really go in detail, figure out what we think is going on with the FATS genes, here we don't really know what's going on. But TBX15 is associated with regulation of production of one of several genes associated with that, of brown fat versus white fat. And as you know, white fat is storage of fat, whereas brown fat produces heat and is thermogenic. And the more brown fat you have, generally the leaner you will be. And there's been a lot of anthropologists have hypothesized that Inuit have more brown fat than other populations. We actually don't know that for sure, but that's been claimed previously in the literature. So we think maybe it has to do with that, but we haven't really done those studies that some complicated or difficulties in getting the kind of samples that we need to really investigate this question. But one thing that we know for this gene, even though we can't really get at the functional side very well, is this gene is in fact also integressed from denisovans. So what these pie charts here show you is the among different haplotypes, which population groups you find them in. And what we have here in yellow is the denisovans, and what we have over here is Neanderthals, and then we have one human group here, and another human group here, including the Inuit, and they are very closely related to the denisovans, and we can show again in statistical analysis that the divergence time between these haplotypes is so short that it has to be transferred by integration from denisovans and into the ancestors of the Inuit. So another example of what we think is an adaptive integration event. Excuse me just for a second. All right, so we have seen this adaptive integration. We've seen that in the Inuit. We saw it in Tibetans, and many other groups have seen it in many other cases. And we think now, when we're going to understand how humans have been so good at going so many places and colonizing so much of the world, part of the process that's gone into that is that there were already other hominins before us in these different areas of the world, and we could pick up genes from them that allowed us to fast adapt to these regions. Moving out of Africa, fast going into Pleistocene, very, very cold regions. Presumably the integration from other hominins that were already living in those regions seems to have been very crucial and important evolutionary event. All right, so I'll give one last examples of this idea about finding mutations of strong effects that are common in the population by looking at positive selection. And this is a study that we did on these Bajau sea nomads that live in Indonesia and other places in Southeast Asia. So this is a project done in collaboration with Eske Willerslev, who's here seen in his natural habitat in front of the cameras, and really his graduate student, Melissa Ilado. So this project is a project that she basically came up with. She had this idea about, she had lived among the Bajau people, and she had this idea for this project. And we basically said that it was a fishing expedition and she probably shouldn't do it, but she was very persistent. So in the end, we said, okay, we'll support you in doing this. And it was good. We listened to her because it turned out to be a really, really interesting project, and teaches us all a little bit about sometimes we should listen to our graduate students once in a while. This was a good project. So what Melissa was interested in was these Bajau people. So they have a very, very unique lifestyle. They live an entirely marine dependent lifestyle as sea nomads living in boats in the ocean. And they get most of the diet from marine sources. They can dive over 70 meters with nothing more than a set of weights and a pair of wooden goggles. And they spend 60% of their daily working time underwater. It's quite extreme. They expose themselves to acute hypoxia again and again and again and again every day. All right, so the question is, what are the phenotypes to look at here? And one of the things we're interested in was what's known as the diving reflex. I'm not sure if you are familiar with the diving reflex, but it's induced by apnea, by holding your breath. and by cold water facial immersion. There's a little test you can do of that where you just ask people to hold their breath and put their head into a bowl of cold water, and that will elicit this response. It's something we think maybe all mammals have. The effects of that is bradycardia, so lowering of the heart rate, which lowers oxygen consumption, peripheral vasoconstriction, again getting the blood into the central organs and the brain where it's needed. And then there's also a contraction of the spleen. Now, does the spleen contract? Well, the spleen you might know for its role in terms of white blood cells, but it's also an organ that holds a reservoir of red blood cells. When it contracts, red blood cells get it injected into the bloodstream. And many diving mammals, they have large spleens. So an elephant seal, a big elephant seal might have a spleen that weighs 100 kilograms. I mean, that's a huge spleen. Okay, and it's thought that it's almost like a diving tank for them. They go diving, and then they get all these oxygenated red blood cells that go back into the bloodstream. So we are interested in looking at the spleen, but as particular spleen size because of this reason. Also because we could do that non-invasively. So there are very few things that you can do completely non-invasively, but you can measure spleen size using ultrasound non-invasively. And here's Melissa down in Indonesia, in Sulawesi, measuring spleen size in one of the study participants. So if we look at spleen size in the bajau, it's in average much larger than some neighboring group, the Saluans, which do not live a marine lifestyle. Of course, that could just be plastic. It could be that if you dive a lot, that induces the spleen to become larger. That's possible. But when you look within the bajau people, on the individuals among the bajau people who dive and who do not dive, we actually don't see a difference in spleen size among them. So that suggests there could be a genetic effect here. Of course, it doesn't show it's anything about genetics, but it suggests, okay, there might be something to look at here. So what we did was we did a selection scan again, and we found our top genes that show the strongest evidence of selection. And then among those, we looked at, are any of those associated with spleen size? It turns out there's one that had strong evidence of selection and also reasonable strong evidence of being associated with spleen size, PDE10A. So what is PDE10A? Well, it's involved in signal transduction related to alpha-adrenergic responses, like the contraction of smooth muscles. Our lead SNP wasn't in the UK Biobank at the time, but SNPs in LD with it was strongly associated with hypothyroidism. In mice, thyroid hormone is associated with spleen size. And we could then go to another database where we could look at the same SNP in another database and confirm that it's associated with thyroid hormone, in fact, in that database. We could also look for spleen size in that database and confirm that this SNP is associated with spleen size, also in that database. Since then, Melissa, what she did for her postdoc was that she made a mouse model where she could actually look at that, and she could show that she could get the same effect in the mouse model, that she could manipulate that gene and lead to higher, larger spleen size also in mice. So she could, in some sense, validate the effects also in mice. So what we want to conclude from this is that we think that selection on this gene, or something near this gene, PDE10A, has resulted in increased spleen size in the bajau people, presumably as an adaptation to diving. All right, so that's the end of my talk. I want to, in the end, start by first acknowledging all the study participants in all of these studies that I've been talking about, and then also, of course, all the members of my lab who has been carrying out much of that research, and then all of you for listening to me here today. Thank you so much.
Great, thanks. We have quite a bit of time for questions. If anyone has one, I can start. So you have these populations. They're quite isolated sometimes. There's a strong selection for a particular trait. How often, or does it is it possible that this trait selection creates a severe bottleneck, and you're actually selecting for familial inbreeding in essence, and so there's a lot of traits that will occur in the population that actually are just by chance, and it's all because of that family just happened to have the one beneficial mutation.
Yeah, I mean, I think generally you're asking the question, these are sort of population isolate, and how do we distinguish bottleneck effects from selection? So that's the whole key here. How do you do that? That's in terms of the statistical methods. And generally, what we're trying to do is in various ways to model that. So we model the effect of bottlenecks, we model population sizes, and we ask the question, is there, with the effect of bottleneck we can find in the population, what would be the variance in allele frequency? How much could the allele frequency change by that? And see, can we reject the hypothesis that it's just simply, a bottleneck in these population isolates? So one of the sort of big questions is always, what do we need to do to show this actual natural selection, right? That's exactly what we're trying to do and answer many of these questions. So, and the key is that you need to be able to model how genetic drift and the bottleneck effects have worked very well to be able to say something about that.
I mean, there is always the just so risk of
absolutely. Yeah.
You create a story in your head for why that trait is selected.
Yeah, absolutely. And that is always, I mean, so I should say, one thing is to show their selection. I think that we know how to do that and can show their selection. But the question is why, and that is where it's much more likely to get at these just so stories, right? We can see that there's individuals that had the mutation, in average, had more offspring than individuals that didn't have the mutation. But why, right? And that is the just so story. For example, with the FATS genes, we know what they do. We know that the particular alleles, we know what they have effect of the enzymatic activity. We can show that. But what has selection actually acted on there? Is it the LDS cholesterol? Is it like of the many highly, these are highly pleiotropic genes. We don't know why some individuals that had that mutation had more offspring than other individuals. That's very hard to say.
Yeah, I have a question over here.
Great.
Yeah. So Charles Rotimi, again, thanks for excellent talk. I appreciated the selection discussion in the Inuit in regard to lipids, but I wasn't quite clear about, it looks like there's also implication for type 2 diabetes, insulin production and secretion. And I wonder if you can share some insight into that. Do they have less diabetes also?
Well, they actually have quite a lot of diabetes, but there's a special story about the Inuit and diabetes. That is, and I didn't show that, some other work that I was involved with, but really led by some of my grad students, they showed that they have a mutation in a gene called TBC1D4, where they have a high frequency mutation in the Inuit that you really only find in the Inuit, where if you have that mutation, it's almost Mendelian if you're homozygous for type 2 diabetes. So where type 2 diabetes is extremely polygenic, in no mutations of strong effect in, for example, Europeans, with enormous amount of work we have put into trying to find the genetic base of type 2 diabetes. There's something we learned is it's highly polygenic. Not so for the Inuit, actually. They have another mutation. This mutation here in the FATS genes may also contribute, but particularly that other mutation really contributes to a relatively high rate of type 2 diabetes on a more glycemic diet, modern diet in the Inuit. So yeah, they are different in terms of type 2 diabetes etiology, the Inuit. I don't know if that answers your question. But these particular mutations, I don't know if we know how much of the risk susceptibility to type 2 diabetes they explain in the Inuit population. So I don't have that for you, no.
Okay, thanks.
Yeah. We have one from online.
So there's one question from an online audience here. So the question is, do you think modern medicine will have an effect on human evolution going into the future?
Yes, so the question, do I think that modern medicine's effect of human evolution? So you often hear about that because we can now treat all these diseases, we survive with these diseases and we produce offspring. So more deleterious mutation will accumulate in the human population. I think that that's maybe the question, the way I interpret it. And I will worry about that in 10,000 years. I'm not particularly worried about that right now. But in 10,000 years, let's look at it again.
Okay, thank you.
Yeah, kind of a related question then. The examples you have, the 3 examples, these are all kind of isolated populations. Maybe that's where you see the positive selection, being reflected. Whereas in the general population, do you have examples of such positive selections?
Well, I think it's absolutely right that you would tend to see these mutations of strong effect in some populations that have recently experienced a change in the environment. Often we find them, in extreme conditions. So Tibetans, extreme conditions up in the Tibetan plateau, the Inuit in the Arctic with that very extreme diet, the Bajau, the divers. And I think what you find is if you, for example, take large European populations, where there haven't been that population maybe sort of average or a longer time, not so specific extreme environments, we don't find so many of these mutations of a strong effect. But I should say that these FATS genes, turns out that they are actually also segregating some of these mutations are also segregating in Europeans and in other populations, and might actually be of more importance there than previously thought. So there might be some evidence of that. And I think maybe we can think of the FTO mutation in Europeans, maybe also an example of this, that might have to do with changes in diet and selection associated with changes in diet. So I think that those would be the best examples in, say, the big European cohorts that people have been looking at. But otherwise, the argument is that maybe there's a lot to gain from studying some of these populations that are not Europeans. Then instead of, if we should put our money on finding new genetic effects, don't put in on making bigger and bigger and bigger European databases, but study many other populations where there actually might be something more interesting to find than spending more billions of dollars on Europeans.
Right, kind of in this regard, sorry, just very quickly, so that do you see similar environmental challenge or pressure that you have different ways to respond? Inuit, you mentioned the fatty acid, but how about Eskimo or other populations living in the Arctic environment? Do they have a different genetic contributions or different ways to handle that?
Well, the Inuit are
they are kind of related.
So we think now that's sort of colonial expression, so we don't use that. Right. But those are the Inuit, yeah. So we know they have a different genetic, right? I mean, they with type 2 diabetes, for example, and so on, yeah.
I mean, you see in similarly extreme climates, but in different populations, you see a different kind of adaptation. Right. Is there someone in another cold climate that
has a different, yeah. So so if we had some other population like the Inuit that for a long time had lived in a cold climate, we don't really have other populations quite like the Inuit. But there are also Siberian populations that are only distantly related to the Inuit that seem to have some other mutations that are also associated with fatty acid metabolism. Yeah, but not quite the same mutations.
Yeah, so I just wanted to respond to Paul's question in terms of whether we see this in non-isolated population and things like that. I think we have an example, the APOL1 variant in relation to kidney disease that is, again, high frequency, almost 40% among West Africans, and about 20% among African Americans. Is highly deleterious to your kidney, but it's absolutely protective in terms of trypanosomiasis infection, the so-called African sleeping sickness.
Yeah.
So that's an example that is,
Yeah, that's good. And maybe I should have said that of course we have some other cases of mutations of strong effect of high frequency, and that's where there's overdominance, like the cases that you're talking about there. And we also have the overdominance cases, of course, sickle cell anemia and so on, beta globin, very famous cases there as well.
Yeah, another question from the online audience. It's called My Call. So, looking across human genes under positive selection, do you often come across candidates involved in the immune system that may be antagonized by the pathogens?
Yeah, I mean, so generally when we do scans in the human genome and try to find selection, most of what we find is on immunity and defense. That is the strongest signal, definitely, it's associated with defense against pathogens. Not surprising if you think about how many, what causes deaths among humans traditionally 10,000 years ago. Pathogens would be very important. So it's not surprising, I think, that pathogens are driving much of the selection we see. The example I talked about was specifically the ones where we see something else, because in some sense this selection associated with pathogens, we almost consider that to be obvious and trivial because we see it all the time. And it's the challenge with that is often very difficult to relate to specific pathogens. We know that the selection on something that's immune defense related, but which pathogen it was, that's almost impossible to say most of the time. We just say these are immunity and defense related genes, and then we don't get so much further with that story.
Very nice talk. I just want to ask, I noticed on your second peak in the Inuit population, you talked about one of the genes, but actually I was hoping you would talk about the other one, which was WARS2, which is kind of interesting because it's an amino acid, it's a tryptophan amino acid synthetase. And I was curious if you had looked into it at all in terms of that population and what implications it may have had.
We haven't, and it's a very good idea. We kind of got stuck with that part of the study, but I'd love to talk more about that.
So deleterious variants in that cause mitochondrial disease. So that's why I was asking.
Okay,
I was just curious.
Honestly, we don't know much more than what I said here, and we sort of gave out on that path. We've decided that we needed biopsies to be able to deal with that, and since this is a study that is more of sort of evolutionary interest, an IP board wouldn't give us permission to get biopsies. So it kind of got stuck on that. I think quite reasonably, well, maybe. So we kind of stuck on that, but I'd love to, maybe there are other avenues we can explore that wouldn't require biopsies. I'd love to hear more about that, yeah.
Thanks.
Okay, great. If there are no more questions, let's give one more hand to Rasmus for a great talk.